Welcome to the 7th annual Midsummer Nights Science and to its 4th and final lecture of the season, the Coelacanth, its evolution, and how fish came onto land. My name is Vivian Siegel. I'm the Director of Education and Outreach here at the Broad Institute, and it's my honor and pleasure to be able to welcome you and to host this series. How many of you have been to Midsummer Nights Science before? Just show of hands. How many of you have been here this year all? Okay, cool. We've got a lot of repeaters. We're actually thinking about expanding our public lecture series to go into the calendar academic year. How many of you would like us to do that? Okay, great. You'll be hearing from us, and so for those of you who've registered online, we have your email addresses, I hope, and we'll be able to let you know about future public lectures when they happen. The series is really about getting you to get a feel for the research that goes on at the Broad, which is a lot about both genomics and also about medicine. Up until this point, we've had lectures almost entirely, or sessions almost entirely on genomic medicine. Today, there will be no medicine, I think. It will be a different topic, but very interesting one nonetheless. So since you know who you are, I won't say who we welcome here, as I always do, but I will remind you that for those of us who actually can't make it to a particular Midsummer Nights Science, we do record these lectures and we make them available on YouTube, and if you haven't already subscribed to the Broad Institute YouTube channel, I encourage you to do that, because some of these lectures that are available to the public happen at odd times at the spur of the moment, and so you might want to catch them on YouTube. We also tweet this series, and you're welcome to tweet if you want to. Our hashtag is BroadTalks. Okay, so we're really excited to have this final speaker, Jessica Alföldi, here today. Compared to our other speakers, Jessica's actually a relative newcomer to the Broad. She joined the vertebrate genome biology group about 5 years ago. Before that, she was a graduate student at MIT working right next door at the Whitehead Institute, where she led the mouse Y chromosome sequencing project in the lab of David Page. So she was actually at the Whitehead at the time that the predecessors to the Broad, the Whitehead Genome Sequencing Center, was there, but she didn't actually get involved with the Broad until she came here. She was the lead Broad researcher on a project that was published earlier this year in Nature, which was on the coelacanth genome. That paper garnered a huge amount of interest in the press, and it was one of the reasons we thought it would be a fun story to tell during Midsummer Nights Science, and so we're really excited that Jessica's here to tell us about that story and its background. So please join me in welcoming Jessica Alföldi.
So hi everybody, I am Jessica Alföldi, and today I'm going to tell you about this amazing fish, my favorite fish, the coelacanth. I'm going to tell you a bit about its discovery and its strange drama-filled history, what we know about the coelacanths and what in fact was known before we even started sequencing it, and about the Genome project, which I am one of the leaders of, and that it was sequenced here at the Broad. So first, the coelacanth was first described, but the fossils of the coelacanth was first described in 1836. Since then, the past couple hundred years, there have been 125 different extinct species of coelacanth fossil that have been found all around the world, all different shapes and sizes, and they're all about 400 to 66 million years old. And so you have to understand that when scientists saw that these fossils stopped at 66 million years old and that the dinosaurs went extinct about 66 million years ago, and they didn't see any coelacanths swimming around, that they assumed that the coelacanths had gone extinct with the dinosaurs, as so many things did. But then, 1938. So this year is Margaret Courtney Latimer. And she was the curator for a small natural history museum in East London, which is a town on the coast of South Africa. And her mission was to actually fill her museum with stuff. She was one of the first curators. And so she had a deal with a friend of hers, Captain Goosen. He was a fishing captain, and he would go out and trawl the coast of South Africa looking for fish, and he would bring her back anything weird he found that she might like to put in her museum. So one day, just for Christmas, she got called down to the docks to check out another catch of fish by Captain Goosen, And she found this amazing fish, and I'm going to read you her words on discovering it. "I picked away the layers of slime to reveal the most beautiful fish I had ever seen. It was 5 feet long, a pale mauvey blue with faint flecks of whitish spots. It had an iridescent silver blue-green sheen all over. It was covered in hard scales, and it had 4 limb-like fins and a strange little puppy dog tail. It was such a beautiful fish, more like a big china ornament, but I didn't know what it was." So Margaret Latimer had a high school teacher who happened to be really interested in fossils, particularly fish fossils. And so she knew that the kind of armored scales you see on this fish, which is in fact the fish that she found, had only ever been seen in fossils. She knew this was something incredible, but she didn't know what on earth it was. So she was determined to preserve it and then find someone who could help her figure out this thing. Now, the situation was this. It was 1938. She was in a small town in South Africa, and she had a 150-pound, 5-foot rotting fish on her hands. So she thought about it. There were only 2 sort of large fridges, like walk-in fridges in town. There was the grocer and the morgue, and they both refused flat out to keep her fish. And so she went to the hospital and begged some preservative, like not a lot, like not enough, but something. And so she, soaked some newspaper in the preservative and wrapped up the fish as best she could. And then she phoned a friend of hers, J.L.B. Smith, who was a professor of chemistry and a sort of amateur ichthyologist. He was into fish. And the problem was, it was Christmas time. He was not there. She could not get hold of him. And so she ended up having to forward a letter off to wherever the heck he was with his family, and she waited. She waited for 12 days and heard nothing. And in the meantime, this fish was rotting. This gigantic fish was rotting. And so what she ended up having to do was she had it taxidermied, which of course she didn't want to do because it means you throw out all the insides. That's what happens when you taxidermy it. And of course, knowing what, studying a fish, you are interested in the insides as well. But it was either that or lose the entire thing to rot. So she had it taxidermied, and then J.L.B. Smith finally got her letter. She'd sent him like little pencil sketches about what the thing looked like and a description. And he looked at them and immediately knew it was a coelacanth. And it completely blew his mind. He called her back and desperately wanted her to try to keep the guts somehow, but they were gone, they were rotten, they were already flushed out to sea with the garbage. And he came over and started studying this fish. In fact, when he said first knew it was a coelacanth, he said, "No living creature has a tail like that." Now, he ended up publishing his description of the fish in Nature, and the first sentence of his paper, which I love, is actually a quote from Pliny, "Ex Africa semper liquid novae," which means there are always strange things coming out of Africa. Papers had style back then. But in any case, he named the fish Latimeria shalomni after Margaret Latimer, who identified and preserved it for science. And Shalomni is after where it was found, the mouth of the Chalumnus River as it emptied into the ocean. And so this got published, and it was like a giant media sensation, both in science and in the public, because you have to understand, for a fish person, this was something like if a Tyrannosaurus Rex had suddenly popped up out of a jungle. People went nuts over it. And so he desperately wanted to find another coelacanth, a second coelacanth, and he would preserve the guts, and you actually get to take a look at what it was like on the inside, and not just the scales and the bones. So he funded expeditions, he put up rewards, but nothing for 14 years. Not a thing. And this thing was a very popular fish. It was a lot of money. People were looking for it. Nothing for 14 years. Until one day he heard from the Comoros Islands. So the Comoros Islands are this island nation. There's a few tiny islands in between Madagascar and the main African coast. And at the time, they were a French colony. And he got word that they had caught a coelacanth. And he was tremendously excited, but also really, really worried, because he was in this situation where there is this dead fish on an island out of his country. It is Christmas again. And he wants to get this fish quickly before it starts to rot. And so he pulled all the strings he could, because he was kind of famous by this point for the coelacanth business, and he got hold of the Prime Minister of South Africa, who, believe it or not, commandeered a squad of the South African Air Force to fly over and get this fish. So J.L.B. Smith got in his the Air Force plane, and you can imagine the poor, the pilot and the crew, they're being called out at Christmas to fly to another country to get a dead fish, which they were not terribly amused. But in any case, he got it, he brought it back, it made his career, he spent decades, he preserved it perfectly and spent decades analyzing the insides and everything about it. But one thing that was very important is he actually found a home of the coelacanths. So the local fishermen there, it wasn't a common fish, but they knew of it. They caught it occasionally, and they called it gombesa. And so now there was a home of the coelacanths. So the thing is that the French government, who controlled the Comoros at the time, were not incredibly thrilled that this major scientific discovery had been flown out of their territory and studied by South African scientists. So they actually put forward a decree that from then on, coelacanths were only for French scientists to study. To be fair, J.L.B. Smith knew that he was doing something slightly underhanded. He did it quick and silently for the very reason that he knew that if he went through official channels, they would never let him have it. But on the other hand, the ethics of whether the French government gets to have all the Comoros fish is a whole other matter. In any case, for many decades, they captured dead coelacanths, obviously, but they tried to capture live coelacanths, to put in aquariums to study. And they had no success at all. You can imagine it would always be a hard thing. That's a giant fish. It's very hard to, capture it and keep it alive. But never a single fish, and it just killed them, and they gave up trying eventually. In the 1960s, they sort of gave up on their edict of only for French scientists, and they sent them around the world to various museums with the promise they had to be used just for display and not for research. And in fact, this one right here is the one just up the street in Harvard's Natural History Museum, which you can go visit anytime you like. But so this, dozens and dozens of coelacanths were sent around to museums, and then after a certain point, people were starting to wonder, well, we don't know how many coelacanths there are. There aren't very many. It's hard to find them. Perhaps it would be best if we stopped catching them now. And so in the 1970s, the Comoros became independent, and in 1994, they signed CITES, which is the Convention on International Trade of Endangered Species. And so now it is extremely difficult to get any coelacanth samples out of the Comoros as well. It should be. But now I'll move on to tell you about something with coelacanth scientists. I've worked on many different organisms, genome projects of various kinds, and scientists, they love what they study, they love what they do, but coelacanth people, they love it more. Lizard people are great, but they haven't been dreaming about lizards since they were small children. Coelacanth people, it's their dream, and one of those people is Hans Fricke, who really want to know what coelacanths were alive. At this point, people had seen dead ones, and they'd seen dying ones, but it really wasn't the same thing as seeing what they were like at home. So he designed and built these submersibles. This is, I think, the second one that he built after the first one didn't find a coelacanth, and he built them specifically to find coelacanths, although he ended up using them for other underwater exploration purposes as well. This one is the Yago, and you can see it coming off a trawler here, so you can get an idea of how big it is. And right here is the video. A video of a live coelacanth, or two, actually. So during the day, they live in these deep ocean caves, often in groups. And they have these amazing glowing eyes that I'll tell you about later. But my favorite thing about them are these fins. They're not like any other fins you've ever seen. They look like limbs, right? They have these stalks on them, and they're known as lobe fins. And they kind of sweep the water instead of just sailing through it. It's really an amazing thing. And those white spots there are unique to individual coelacanths. People can use them to identify coelacanths when they come back later. And there they are at repose in the water. And other people who are very much into coelacanths, Laurent Balesta. So he's an amazing underwater photographer and biologist who does these expeditions down to find coelacanths now. This one is in South Africa, and you can see sort of the scale of the thing right here. I'd recommend you check out his websites, entremedocean.com and his coelacanth one to see some truly beautiful, beautiful photos. He actually uses special equipment to be able to go down much further than your recreational scuba divers can. So he can actually take much better pictures outside of a submersible. So the second part of the story of the coelacanth comes quite a bit later, in 1997. This is Mark Erdman. He's an American marine biologist, and he was graduating from grad school and was going to move to Indonesia for a postdoc. And in the meantime, he was getting married in Indonesia, had a lovely destination wedding and a honeymoon. And he was in a town called Manado, and he was walking through a fish market when he saw an old man pulling a large fish. And he looked at it and was like, well, that's a coelacanth. And he was really excited, but when he stopped for a second and said, wait, I mean, he was a marine biologist, but he's not a coelacanth expert. He said, well, maybe people already know that there's a coelacanth in Indonesia. Maybe this isn't a big deal, and I would just be making a big deal out of nothing. And you have to imagine his situation. He's in a town in Indonesia he does not know. This is a 150-pound fish. He is on his honeymoon. What is he supposed to do in this situation? So what he ended up doing was taking a picture and letting the old man go with his fish to sell it in the fish market. And he got back home and showed the picture to all of his biologist friends, and they all freaked out because, of course, no one had ever seen a coelacanth outside of a small area on the African coast before. And so he went back to Indonesia right away and started, expeditions, rewards, the whole deal, trying to find more coelacanths. And he ended up, after 6 months, finding actually the original old man, believe it or not, on a small island called Manado Tua, just off the coast of Manado, where they do occasionally catch coelacanths. They call them Raja Laut, which means king of the sea. And he named this fish Elatomira manadoensis, a different species of fish, after the location where it was caught. But the drama continued. He published his discovery in Nature. But then a couple of years later, a group of French scientists in Indonesia came out and said that we had actually discovered the Indonesian coelacanth several years ago, but we had lost all the evidence, and now we've just found it. Unfortunately for them, of course, that there was easily available software by this time that could show that it was actually a rather clumsy Photoshop job, actually using the very image that he had published in Nature. And you can see sort of the end of this news article here. Admits that the two photographs do appear to show the same fish. This is very embarrassing, he says. So once the second species of coelacanth was found, then people started descending on Manado. Partly obviously out of excitement, a second species of coelacanth, but also because with CITES, there were restrictions on the African coelacanth, but they're specific to a species. So right now, there actually were no restrictions of any kind on the Indonesian coelacanth. So scientists came to study it, aquariums came to try to catch a live one again, because I'm sure every aquarium would love to have that as a prize subject. But good for Indonesia, they immediately declared the fish part of their national heritage. It could be restricted, and now it is on CITES regulation and is very well preserved and hard to catch once again. If you're interested in these kind of stories about the discovery, I recommend this great book by Samantha Weinberg, A Fish Caught in Time. There are actually even more crazy stories about the discovery of the coelacanth. I just don't have time to tell them all. So now to move on to what we actually know about the coelacanth, what it is, what it does, and the sort of things we knew before the Genome Project started. So where does the coelacanth live? It lives in these ocean caves. They're about 100 to 500 meters deep, so just a bit too low for like normal scuba diving, I would say. This is the deep ocean, but not crazy deep, not the great depths of the ocean, just deep enough. They come closer to the surface at night, not to the surface, but further up to hunt and to eat. And we think we understand now why they're so hard to catch alive. And it's just that, they're in tropical oceans, that the surface is very warm, but where these guys live, in the deep, are actually pretty cool. And their blood oxygen is adapted to that kind of temperature. If they're kept up top, in the heat for any length of time, they sort of gradually suffocate and die. And this means that it will always be very, very difficult to capture alive if anyone decides that's allowed again, because you essentially need like sort of a gigantic refrigerated aquarium box, and I don't even know if that's possible. So where have we found coelacanths? So right here is the one spot the Indonesian coelacanth has been found, the island of Manado Tua. And African ones these are all the spots they have ever been found. I mean, this red dot is Margaret Latimer's first coelacanth. So some of these dots are just sort of single findings, but some of them are actual communities that are known to exist. Like there's a population off the coast of a national park in South Africa. Here are the Comoros Islands, and there's also a population off the coast of Tanzania. But some of these others might have just been drifted away by the currents. We're not even sure. And the thing is that most coelacanth scientists will tell you that the odds are low, that these are the only 2 pockets in the world where you find these things. They're just very unlikely to be found or caught. It's too low for scuba diving. You'd have to be doing deep sea fishing at night, which the people who live near this island do, and the people who live in the Comoros do, but isn't a common thing in any case. And so I would not be surprised if there are pockets of these in caves in various places around the world. It's just unlikely that we will find them, and if someone found them, that they would know what the heck it was. So how does a coelacanth move? Here's a close-up of one of those really cool fins. They're these limb-like fins on stalks that sweep the water. And one neat thing about them is that they're used in pairs, like land vertebrates. Like if you see a horse move, they have like the opposing limbs that move together. And it's the same deal with coelacanths. How does the coelacanth see? So the coelacanths have these glowing eyes, the live ones do. In the water, they glow blue. But the fishermen who have caught them have said that when they catch them at sunset, they glow like bright coals, which I thought was pretty cool. And the reason why they do this is they have an extra layer behind the retina, the tapetum lucidum, that reflects and intensifies light because there isn't much light down in the depths where they live, and this helps them to see a bit better. If you've ever seen cats with eyes that glow at night, it's the same kind of idea. The coelacanth is almost colorblind. It just has a few of the cones in the eyes that we use to see color. So it probably sees like really washed out color, but that also makes sense because there isn't a lot of light and so not a lot of color to be seen down there. But the coelacanth has another way of seeing things, and that is through electrical fields. So there are many fossils that show this space called the rostral organ in the nose, but the only living thing that you can see one in is the coelacanth. It's filled with gel, and it detects electric fields, and is believed, although it's not like we can do experiments on the coelacanth, that there's something in the tail that generates an electric field, and that the nose then senses them. I mean, I guess analogous in some way to bats and sonar as a way of like seeing its world. And one thing you can see, well, you can see the fins moving in pairs here, which is kind of neat. But the coelacanths, quite frequently, you'll see them go really vertical, head down, which is something I have never seen any other fish do. I don't think anyone has ever seen any other fish do. And so there's a neat little video showing what it does. You can see it fully extended there, head down at the ground, and then the tail going straight up. And they'll stay like that for, hours at a time. They do seem like they have too many limbs, don't they? So how does the coelacanth eat? It's another terribly strange thing with the coelacanth. It has something called an intracranial joint that lets it unhinge its entire jaw to capture its prey. And once again, this is a thing that you only see in fossils and in the coelacanth. It has teeth, but not terribly impressive teeth, as you can see. They actually don't bite or chew anything. They're just to prevent the prey from escaping after he's gone in his mouth. It eats a wide variety of seafood, your squid, your eels, your skate, and up to 3 feet long have been found in the stomachs of coelacanth. They're essentially just swallowed whole. This has never been seen, but I imagine it'd be quite a sight. So other variety of strange things about the coelacanth. So in vertebrates, and most vertebrates in general, embryos that are developing have something called a notochord instead of a spinal cord that's eventually replaced with the spinal cord. The coelacanth has a notochord all the time. It never makes a real spinal cord. We don't know why. The skeleton of the coelacanth is mostly cartilage, like the stuff in your nose, not like actual bone. They give birth to live young instead of laying fish, instead of laying eggs, as most fish do. And my favorite weird fact about it is its tiny, tiny brain. So the coelacanth is about human size, 150 pounds, 5 feet. Our brains are about 3 pounds. The coelacanth brain is less than 1/10 of 1 ounce. It has the smallest percentage of its weight devoted to its brain of any vertebrate alive today. It's found all the way at the back of its skull cavity. Like, it has sort of a normal skull cavity, but it's all filled with fat. And these days we know that within a species, like the size of your brain doesn't mean you're more intelligent. And they're even known that songbirds, they're small, they have small brains, they have complicated behaviors. But the percentage of your body devoted to your brain is sort of generally thought to be a proxy for your intelligence. And so the odds are that the coelacanth is not the brightest fish one has ever met. So now, before I tell you about the coelacanth project itself, I'll tell you a bit about why we do this at all, why we sequence genomes. So this here is a picture of the phylogenetic mobile that you can see out in the lobby, actually. And this is a couple years old now, but when it was built, it was a description of all the placental mammals that had been sequenced. And so that's not including the marsupials and not including your birds and your lizards and your amphibians and your fish and your bacteria and your plants and your viruses, just the placental mammals, and it's quite a few of them. And so the question is, why another one? And the reason is that there's sort of twofold reasons. One of them is that for scientists who work with a particular species are very excited to have a sequenced genome. It saves years of work. It lets you do things in a couple weeks that would normally take you years. It makes whole avenues of work, of research possible. But it's also a resource for scientists who study evolution in general. People are interested in how evolution works in sort of a broad sense, the different ways it works over large scales of time. And there are people interested in specific functions, smell, or how the body deals with sugar. And you can see how that is done in various different animals. And from that, you understand how evolution has worked on that one feature. The question is, why sequence the coelacanth itself? And the first answer, I would hope you would understand after my talk, is that it is incredibly cool. And although we have a large number of very valid reasons, scientific reasons why we should sequence the coelacanth, I think deep in the hearts of coelacanth people, it is first and foremost because it is incredibly awesome. But the two main sort of scientific areas that we wanted to address was, first of all, this question of slow evolving. And so this is a difficult question. So when J.L.B. Smith first wrote this paper, he called the coelacanth a living fossil. And what he meant by that is that there are all these fossils of coelacanths, and look, here's one alive. But a lot of people over time have taken this to mean a wide variety of things, including that, that the coelacanth doesn't evolve, that it's actually our ancestor or something crazy like that. It's not our ancestor, it's a currently living fish. Which is why evolutionary biologists get a little bit nuts about the phrase living fossil. But the question remains is that while there are a whole bunch of paleontologists who say the coelacanth looks a lot more like its fossils than other things do, maybe it's slow evolving. And then there are other paleontologists who say, no, you're crazy. It doesn't look much like its fossils compared to other things. It's kind of a subjective thing that is hard to deal with. But in any case, we want to see if we could actually finally address this question in sort of a real quantitative way. We look at the genome, is the coelacanth's genome slow evolving? And the second sort of general area is, how is the coelacanth related to the first fish that came up on land and eventually became us? And more generally, how we can learn about the process of adapting to a new environment like land. Like how did we change to be able to deal with land? So the first step was that the thing is, everyone knows the coelacanths are incredibly endangered and incredibly rare. And for anyone to agree to embark on a project like this, they had to be convinced that you could actually get a sample to work on. And so a group of scientists in South Africa, including Rosemary Dorrington, determined that they had to get a sample first before anyone would ever agree to even work on this thing. So in 2002, which actually not long after the human genome got sequenced, really, she flew off to the Comoros and did heroic acts of bureaucracy to get the permits to essentially, she gave equipment and she gave training. And the deal was that they would catch one more coelacanth. The people on the ground would take blood from that coelacanth and they would be able to preserve it. She gave all the equipment and the training. And then she got permission for them to send that blood to South Africa, where her university was. And it worked marvelously. I mean, it had all sorts of problems because, to the Comoros Islands' credit, they had stopped catching coelacanths. I mean, it helps that it's terrible to eat. Apparently, it causes intestinal distress, let us say. But in any case, they'd stopped catching coelacanths, and they even trained fishermen how to avoid accidentally catching the coelacanths. But they get permission to catch just one more, and the blood was transferred to South Africa. And from there, another barrel of paperwork was done to send off to the United States to a lab professor called Chris Emamia at the University of Washington. And he put together a proposal to have this sequence. Now we show that it was actually technically possible. And so from there, the blood came here to the Broad Institute to have its DNA extracted and become sequenced. So the next step was getting together a team. This here is the Coelacanth Genome Consortium. Our paper has 91 scientists on it from 40 different universities and institutes from 12 countries on 6 continents, every continent except Antarctica. We managed one in-person meeting of most of the people in about the 2 years that the analysis took, but you can imagine it was somewhat difficult, since even phone calls are somewhat difficult when you have so many time zones to work with. But anyway, the first step, of course, is sequencing it. So how do you sequence like a 3 billion base pair genome like the coelacanth? So a base pair are like the letters, the A, G, C, Ts. There are about 3 billion of them in our genome and about 3 billion in the coelacanth. And they're in the cells, they're in chromosomes, of course. And each of those chromosomes are maybe 50 to 250 million base pairs long. And the problem is that the machine that most people use to do such things can only sequence 100 base pairs at a time for sort of chemistry-related reasons. And so what you have to do is you sequence a lot of 100 base pair chunks, or reads we call them, and then have to put them back together as a sort of mega puzzle from hell. There are people, very, very smart people, some of the best of which work here at the Broad, and their job is to come up with new computational ways to deal with this problem. And one of the ways we use to help them is something called paired end sequencing. So you can imagine that sort of the first obvious way of doing this is to cut the DNA up into 100 base pair chunks and just sequence them. But what paired end sequencing is that you cut into larger chunks. So these are 4,000 or 40,000 base pair chunks. And then you just sequence 100 base pairs from each end. And I'll show you in a second why this helps. So this is the process of assembly. So these little solid black bars are the bits you've actually sequenced, and the dotted line are the parts you have not. So you can take all the little black bars, and the ones that overlap you can put together into this long contiguous sequence that is known as a contig. But sometimes what happens is that you will have one end of these pairs that is in one contig and one end is in the other, and then you could say, hey, these 2 bits must be next to each other, and I can make sort of a vague guess at even how far apart they really are. And this is what we call a scaffold. A scaffold are a bunch of contigs joined together by information. And this actually is what a genome assembly looks like. It's what even the human genome assembly looked like once upon a time. It was a draft assembly, although they've been working, of course, for many, many years to fill in a lot of these gaps. But the vast majority of genome assemblies look like this. We have most of the sequence, we have a lot of the sequence, but we don't have all of it. And now, before I move on to what we actually learned when we analyzed this sequence, most of it's been based around phylogenetic trees. And so I want to use a family tree to explain to you about how we understand phylogenetic trees for research. So this here is the Weasley family tree. James here and Lily are brother and sister. And if you want to get from James to Lily, you go up to their common ancestor, who is their parents, and then back down to Lily. On the other hand, you want to get to James to Hugo, you have to go up even further to their common ancestor, and then back down to Hugo. So these people are more closely related than Hugo and James. Now here's a phylogenetic tree, which is, in this case, a tree of a whole bunch of different species, how they are related to each other. These are, of course, not all the species in the world, or even the ones that have been sequenced, but they're the ones we looked at for this. And here is us, with the humans as represented by Margaret Latimer. And the closest species that we looked at here was the mouse. So you can see they're like sister species, and if you go up here, that is their common ancestor. If you go out even further, you can get to the dog, and here's their common ancestor. So in this tree, here are all the mammals. Right here, even the more diverged ones are marsupials and your platypus. If you go even further, you end up in the reptiles. And yes, birds are reptiles. You heard it here. And then if you go out even further, you end up with amphibians, represented by these frogs. Now, all of these species together, the frogs and the mammals and the reptiles, are called tetrapods. These are the land vertebrates. They're called tetrapods because we have 4 limbs, tetra. And here is, right here, somewhere in this line, is the fish that crawled up on land and became us. If you go even further out, the question was always, well, what is the fish that is most closely related to that fish that first came up on land? And there was this huge debate for a long time. Is it a lungfish? Is it the coelacanth? Is it the lungfish? Is it the coelacanth? It's the lungfish, we can now say. Some people are disappointed. I'm not terribly, because the lungfish is huge, huge genomes. It's extremely difficult to sequence. No one's going to do it anytime soon. So the coelacanth is still the most useful thing for actually trying to understand this process. So the coelacanth and the lungfish and all the tetrapods are lobe-finned fish. So you and I are both lobe-finned fish. And I hope this makes it clear. The coelacanth isn't our ancestor. This thing is our ancestor. The coelacanth is a living thing today. If you go even further from the lobe-finned fish, that's where you end up at ray-finned fish, which are the things that we pretty much just think of as fish. Any fish that you name off the top of your head is a ray-finned fish. And you go even further, you end up with cartilaginous fish, which are the sharks and other such things. And so we want to ask the question, is the coelacanth slow evolving? And after we posed this question, we realized we don't even know what this question means. What does it mean for a genome to be slow evolving? How would we recognize it if we saw it? And so we took a fair amount of time trying to understand how to pose this question in a way that was conceivably answerable. And one what we came up with was to look at proteins in each of these species and say, how many changes have there been in equivalent proteins in each of these species? And that's actually what this tree shows. The length of these horizontal lines is proportional to the number of protein changes in the few hundred of proteins we looked at. And so from this, you can see that the mouse had a lot of changes, the frogs have a lot of changes, the coelacanth not so much. And in fact, through various statistical methods, we showed that the coelacanth, their proteins have significantly fewer changes. Their proteins are slower evolving in that sense, which I thought was pretty awesome, actually. But the thing is that proteins are translated from genes. Genes are the recipes to make proteins. Genes only make a very small percentage of a genome. What about the rest of the genome? Is that also slowly evolving? And every other method we could come up with, the genome did not look particularly slow or fast or different in any way. The genome is not slowly evolving, but the proteins, and by extension, the genes, are slowly evolving. So let me move on to the other question, the idea of how do we understand about emerging onto land? Now, right here is a picture of Tiktaalik, well, an artist's imagining of Tiktaalik. It's a fossil, an extinct fossil, which is found in the Canadian Arctic. And it is what they call a fishapod, like a fish in the process of becoming a land vertebrate. And the question is, how did that happen? How did those change happen? And how people generally do this is they compare the species that did come on land with a closely related one that did not come on land, and that would be the coelacanth in this case. And so we looked at which genes were lost when we came up on land, and we also looked at regulatory elements. So regulatory elements aren't genes, but they're the things that determine when and where a gene is turned on to make protein. And so we looked which genes changed where and when they were used between us and the coelacanth. And so what was nice is we found some things that you would absolutely expect. The first one being limb development. When I say development, it means like how things are made in the embryo. And you'd absolutely expect that how embryonic limbs are made in you and me is very different than how fins are made in the coelacanth. Other things that were found are eye and ear and brain development. Kidney development was also expected because fish essentially just pee in the water all the time and it just drifts away and it's fine. We, of course, keep our urine in a bag that we carry around with us and get rid of every once in a while, and kidneys have to be very different in order to be able to handle that sort of thing. But my favorite thing is when you find something that you hadn't thought of, but that makes total sense when you think about it. And one of those is smell. We found that genes having to do with smell changed a lot in this process. And that makes sense if you think about it, because the chemistry of a molecule attaching to your nose and being able to smell must be completely different in water than in air. I mean, I don't know about you, I cannot smell anything in water, but fish do. And that got changed. Another thing is immunity. there are a lot of immune system genes that show changes, and that's, of course, because there are different bacteria and parasites in the water than on land. It's a completely different environment. So next I want to tell you about my favorite kind of genomic study, which is the kind where you actually show at a molecular level how evolution works. So first, a little bit of background. I'm going to tell you about a little story about how fins became limbs. So tetrapods have 3 regions in each limb. So you have the stylopod, which is like your upper arm and your thigh, and then there's the zeugopod, which is your lower arm and your calf, and then the autopod is the hand and the foot. Now fish, including coelacanths, they just have the 2 regions. They do not have hands and feet. They don't have autopods. And so people have been arguing for generations, how did the instructions to make a hand come about? How did that happen? Were they adapted from instructions how to make the rest of the limb? Or did they or adapted from something else completely different? And so there have been 20, 30, 50 years of papers on this sort of thing. And we use the coelacanth genome to address this by also taking advantage of an organism about a lot more of which is known, i.e. the mouse. So it is known that there are these regulatory elements. Those are the things that tell you where and when a gene gets turned on. They're known to be absolutely required to make an autopod. If you don't have this, you don't have hands and feet. So we looked at the coelacanth genome and showed that there's a similar sequence for one of them in the coelacanths, but not any of the other fish. And that was interesting. So they took the coelacanth sequence and stuck it in a mouse embryo and said, well, what does this thing do? And they attached it to a sort of gene so it makes blue dye. Anytime that it tries to turn a gene on, instead of turning on whatever it does, it turns on blue dye instead, so you can see what it's doing. And here are some mouse embryos, and you can see, if you know your mouse embryo anatomy terribly well, that it turned it on in limbs, in embryonic limbs right here. Here's a close-up of a limb. You can see that it's turning on. And this means that in this particular case, you need more than this one regulatory element to make a hand. It's a whole slew of things. But this one case was adapted from making a fin. That it took, the genome used this thing that was used to make a fin and used it to make a hand, at least in this case. So the future of the coelacanth. So there are many other things that we published in the coelacanth genome paper. This is just a few of them. And this is just the beginning for coelacanth genomics. The point of this is that we create a resource that all sorts of people can use for their own particular ways. And there are a bunch of people, of course, already working on this. The question of what do those new or changed sequences in the coelacanth do? And people take them and they put them in mice and zebrafish, as I mentioned before, to try to understand what the heck they'd be doing in the coelacanth, because experimental coelacanth biology is really not going off the ground anytime soon. Then there's a question about why are the proteins slow evolving, which we really don't know. And you can examine this by looking at which ones are slower, which ones are faster. Does this give you any clue about why this would be going on? There are people interested in how all these coelacanth populations are related. We know the coelacanth is endangered. We have no idea how endangered or how many there are, really. They're just hard to find. And there are more expeditions to learn more about behavior, because we know so incredibly little about their behavior. We don't know about reproduction or territory or raising young or anything like that. And of course, there'll be more crazy stories of fraud and intrigue, because that's just how things work. So, I just want to thank the 90 other people that I'm taking credit for today, starting with the people at the Broad Institute who made this possible, my friends in the vertebrate genome biology group, Ian and the computational R&D group, who were the people who put the puzzle that is the coelacanth genome back together, and the labor of love it was. Marcia, Joshua, and Lynn, who dealt with some very difficult and rare samples and got some great results. The Genomics platform, who did all the sequencing, and my bosses, Federica and Shostin, and of course, Eric Lander. I also like to thank the entire coelacanth genome analysis consortium, a great bunch of people, especially Chris Amamiya, the other major leader of this group, who has been dreaming about the coelacanth genome project since I was in high school. Axel and Teresa, who worked on the slowly evolving genes. Rosemary, who got that crucial sample. Elve and Henner, who made the beautiful tree. John and Ingo and Venki and Allison, who examined what happens when fish come on land. And Igor, Nicholas, Neil, and Cliff, who did that lovely molecular experiment in mice. Thank you.
Thanks, Jessica. That was awesome. We're ready to take questions now, and I'm holding my phone to remind you there are 3 ways to ask questions. You can raise your hand, and I'll get a microphone to you, and I see the man there I'm getting to in a minute. You can also tweet them to Broad Talks or email them to midsummer@broadinstitute.org.
Please speculate about the possibility of locating another area of the world other than the 2 we already have where the coelacanths are found. And should we read tomorrow in the New York Times that they've found a 3rd area, speculate as to where it might be?
Well, that's a difficult one, I'm afraid, because I'm not a marine biologist. I'm a genomics person. But I mean, rocky caves, 100 to 500 meters deep. It's the same for both of them. Also, I mean, it just so happened that the people who fish in both places use the exact same fishing methods. I would suggest people use those fishing methods elsewhere if they wish to find coelacanths. Hi.
Hi, that was a really nice talk, very interesting. So going back to the figure where you described I guess the proxy for the rates of evolution over time, were you guys, what results were you expecting? Were you expecting the rates of evolution across different species to be the same, or were you expecting there to be significant differences?
Oh, I'll say even within our group, people were betting all sorts of different ways. I have to admit, I was betting that it wasn't going to be very slow. I thought it was improbable that, it's not like in a magic time bubble at the bottom of the world. It's a real organism living in the real universe, but the proteins are significantly slower. They really are.
Hi. Since the coelacanths have a tiny brain and it doesn't have a spinal cord, does it have a central nervous system?
Well, I mean, it has to have a central nervous system, I suppose. It's so big it can't get away with not having one. We just don't understand how it can get away with having a notochord for so long, or why that would be helpful in some way. It's just really unknown.
See if I can find another question. That took a while. Fall. Okay, I'm coming to you.
Is there any, it's kind of a 2-part question, is there any estimate to the size of the population of that fish? And if a population consistently through the ages has been very small, wouldn't that slow down the transmission of the evolution in terms of the SNPs and the handing on?
Well, I mean, small population, we don't know the population. We think it's small, but it's hard to find them. So we can only have sort of like a lower bound. It could be much larger. And we don't even know which. It's thought that the Tanzanian population, African ones, is actually somewhat separate. There's been like a little bit of sequencing done that makes people think that that has actually been separate for some time, even though it's the same species. Yes, a small population might explain it, but it's not enough, was what I think anyway.
How is the slow rate of evolution of the proteins related to how these critters live, their lifestyle, shall we say?
Well, I mean, this is where I don't want to get too far ahead of myself. I mean, I can speculate all the livelong day. I mean, it is possible that if they don't have to adapt that much, that their proteins would need to change, and maybe they don't have much in the way of predators, or that sort of thing. Or they've been in an environment that's not changed that much. That being said, the Comoros Islands are volcanic islands. They haven't been around for geologic time. That has changed somewhat, but an unchanged environment is one way that you might have slower, I guess it's what's called positive selection, the changing of proteins.
Apropos of the first question, where to look next, two very odd things about the two places where they have been found. Geologically speaking, they were, of course, 300 million years ago, part of the same coherent plate. The Madagascar and Indonesian environments have gone their separate ways. The irony is that you mentioned that the fishing methods are the key to finding the things. Well, of course, the Indonesians moved to Madagascar 1,000 years ago.
Oh, yeah. Well, maybe that's how it passed from there to there. You obviously need to get them to move to more places so they can fish up more coelacanths.
How does the slow rate of protein evolution and the proximity or the similarity to fossils compare with sharks? Are they similar to their fossils, and are they any slower than normal in protein evolution?
Well, looking like your fossils is the most subjective thing on Earth, and so you can get even two experts in the field will argue about it all the livelong day. And I am far from an expert in the field of shark fossils. I really can't say.
All right, we had, okay, I'll get to you, and then there's one behind. Oh, that was just one of you. All right.
You indicated that the non-protein coding regions evolved normally, I guess, relative to other species. What about the regulatory elements within the non-coding regions? Do they also evolve normally, or are they unusually slow?
We did not see it abnormal. It was within normal ranges. I mean, that being said, like the resolution for that is not terribly high. We don't have a lot of examples, so something has to be really out of whack before we can tell that is abnormal, and that tells you how out of whack the proteins are.
Is it possible to sequence the fossil and compare and contrast the DNA between the living fish and the fossil?
I wish it was. I mean, fossils, unfortunately, are like sort of negative impressions in rock. There isn't stuff there anymore. I mean, there's been remarkable amounts of success in sequencing old bone, for instance, which used to be thought to be impossible. But we're talking about our ancestor, like the fish came on land, that's 400 million years ago. And the Neanderthals people are sequencing are maybe a couple 100,000 years ago. So it's, there's no Jurassic Park for us this yet.
You mentioned that there's a whole raft of changes that need to occur. It's not just limb development to have vertebrates come onto land. What's your sense of the time that elapsed from the advent of marine invertebrates to land invertebrates, and how does that compare to the emergence of marine vertebrates to when vertebrates finally did come onto land?
Well, it is my understanding that invertebrates came on land first, insects and stuff, and of course plants came on land even earlier. But I'm afraid I don't know the exact timing.
What exactly is the difference between a notochord and a conventional spinal cord?
Notochords are found in embryos in us, and they're replaced by spinal cords. There isn't a ton known about notochords. They're a long rod-like object, and I'm afraid that's the total of what I know about notochords.
We have a question coming in from Twitter. Can you speculate the age of a 150-pound coelacanth?
Well, it would be total speculation, Twitter. Large things, large animals are known to live long. On average, that sort of thing. But no one knows. That's part of what we have no idea, that we have no way of knowing about how endangered they are, because how many kids they have, how long they live is a big part of it. It's not quick. They're not like goldfish.
Yeah, I'll try to remember. It was a great talk, thank you. And I wonder if you can explain this leg motion in fins. It didn't make any sense. I've never seen it before exactly as though you had legs, and you would anticipate some paws or something there, but it's not there. It's the fins moving like legs.
It sort of seemed like sculling to me. When the first coelacanth was discovered, and like people would place them in museums like as if they had legs and were out walking. In the wild, they don't walk on the ground. It's not like that, but they kind of scull. I think of it as if you ever, kayaked, that kind of thing. It moves them through the water just great, but then, of course, other fish do just find their own way. Oh, I've been told, although I have not seen it, of course, that they can go fast when startled. But anyway.
I have a two-part question. One, is it considered definitely known or just probable that humans evolved from fish? And the second part of the question is, capturing and then letting go a coelacanth, you didn't say whether that would cause them to die. What about tagging them electronically?
Okay, well, the first part is that, yes, of course, our ancestors are not the current fish living today, but that once upon a time, yes, we were in fish before we were on land. We were fish before we were on land. And so the second part, tagging, people have thought about this. I mean, there was a campaign before they got the Comoros people to entirely stop accidentally catching them, an idea of finding a way of getting them back down fast enough, but no one actually has any idea whether that works. They maybe were stunned and died and no one ever knew. So they really focused their efforts on just not catching them at all.
Well, if they were tagged, they'd know whether or not they survived.
It's true. I mean, that is something that could be done, but people are reluctant to do. I mean, it's like trying to catch them for aquarium. It might kill them. It's just people are erring on the side of not doing anything, I'm afraid.
So I'm going to pass this along for one last question.
So why did the coelacanth stand on its head?
I don't. Why did the coelacanth stand on his head? I mean, strictly, I don't know. I mean, we do really think it is something to do with the electrical field. I mean, Hans Fricke, the submersible guy, would like emit electric fields and see it do that thing in response. I mean, apparently it makes it easier for them to sense the electric fields, maybe something with the alignment of the tail emitter and the head sensor, but you would have to ask a coelacanth.
Well, from the nature of the questions, I'm sure there'll be a lot of conversation to be had outside in the lobby during the reception. I'd love to have you all join us there. But before you go, I'd like to remind you to please fill out our survey if you haven't already done so. We do rely on your feedback to make this program better, and especially for those of you who'd like us to keep this program going in the year, giving us ideas of what sorts of things you'd like to hear about would be phenomenally useful for me. And so thank you very much for coming, and let's thank Jessica Alföldi one more time.